Every day I get inspired when I come to work because of the questions we are addressing at work, no? That keeps me going every day and motivated. Inspiration at the beginning, I guess it was the teachers at school, teachers from the physics and chemistry. At the beginning I was very interested to be a chemist and I was obsessed with chemistry until my teacher from biology convinced me, I guess, that both things could be integrated. I'm Elias Barriga. I am the group leader here in the Mechanism of Morphogenesis lab in IGC. I am originally from Chile, from the very south of Chile. Our lab is interested to study how the developing embryos acquire their shape, and for that we study the collective migration of cells within the embryo, and sometimes also in regenerating tissues. My interest in science started in the last years of school. I will not say that since the very beginning I was meant to be a scientist and so on, but then I got increasingly interested in this world of chemistry and then biology and the integration of both. Then I moved to the university, Universidad Austral de Chile. There was a professor in that university called José Núñez, and he infected my head with developmental biology. And at the time I moved from Santiago to London, and then that was a snowball that took me to the lab of Professor Guillaume Charras, and then I was interacted with other people from Cambridge, and all that set me from looking to little embryos and things under a stereoscope to start using other type of devices, more from the physics world, to understand how the embryos will acquire their shape. A friend of mine, she told me, hey, as you are looking for this position, there is this place in Lisbon, and it's called IGC, Instituto Gulbenkian de Ciencia, and they are offering this position. And I have to say that when I came to the interview, it was a Friday, and it was a very vibrant atmosphere outside in the patio. And I said, well, this looks like the kind of place that I would like to work at. So I left IGC at the time thinking I want to work there. We address quite a complex question, which is how individuals achieve their shape. So how we acquire form. Within the embryo, there are many things going on, like electric fields and mechanical stimuli and so on. So all these cues, externally, they have been shown to drive directionality of cells. But nobody knows, first, whether they operate in the native context of the embryo, or whether or the way in which they will integrate with this known chemical guidance. I thought, well, this is an interesting question, nobody have addressed it. And that's how, with some motivation from my colleagues from IGC and so on, I decided to put all these questions as part of an application for the ERC fund. Addressing this type of questions is not easy in many layers. So first, it's technically challenging to get to measure these mechanical or electrical inputs. And as it is technically difficult, this comes with at a high cost. What happens there is that you cannot really buy all the machines or microscope or sophisticated equipment that we need to measure and modify electrical and mechanical inputs. The ERC really set us with all the equipment that we really need to address this challenging questions about the mechanoelectrical interplay in guiding the directionality of cells within the embryo.